everyone. Welcome to my on-demand presentation. My name is Guus Kortman. I'm Senior Project Manager and Scientist Microbiomics at NISO, a contract research organization based in the Netherlands. And today I would like to share with you an overview on the role of the gut microbiota in a good night's rest. So to know what to expect, I will first go into the importance of sleep and then the known associations of sleep with the gut microbiota, dive a bit into the mechanisms in the microbiota gut-brain communication, and then the evidence for effects of pre, and postbiotics in human trials, and screening of next generation candidate probiotics. So a good sleep quality is of crucial importance. It's necessary for our physical and mental health and quality of life. And insufficient sleep can lead to increased incidences of cardiovascular morbidity, for example, high blood pressure or coronary heart disease. There's diabetes, obesity, derailment of cognitive function. So in this figure, to name a few, to be easily frustrated, irritable, poor attention span, poor motivation and organizational skills. There's even depression and traffic and workplace accidents. So it's quite serious side effects. And the prevalence of insufficient sleep and daytime sleepiness is globally on the rise. Sleep problems prevalences range from 15 to 30%, depending on the country and the years it was reported over. It's really a sign of modern society and our lifestyle, but there is an urgent need for new interventions that support a good night of sleep because of all these serious side effects. And there is the obvious that we need to reduce stress, the use of electronic devices, and on the other hand, for example, increase physical exercise, etc. But one strategy with great potential is also modulation of the gut microbiota. And I will explain you why. So multiple studies have shown an association of sleep quality with the gut microbiota in both humans and animals. And there is growing evidence for a positive correlation between sleep efficiency and diversity of the gut microbiome. So as you might know, high diversity of the gut microbiome is considered beneficial. And now it seems that we can add sleep quality to this long list of the things that are thought to be correlated with a high diversity. Good sleep quality in humans is, for example, positively associated with, to name a number of taxa here, and members of the gut microbiota, higher relative abundances of microbia, lentisfera, also of Blottia and Ruminococcus genera, and on the other hand, lower relative abundance of Prevotella genus and Lactobacillus family. So it's not really, things that are reported is not really limited to this list, but to name a few and that appear maybe a little bit more consistent, but I will also mention that in a second. But sleep quality, is it a cause or outcome of gut microbiome alterations? So the chicken and egg story. There are studies shown that partial sleep deprivation can alter the gut microbiome composition in already as little as 48 hours. But in contrast, in another study, longer periods of sleep deprivation did not have any effect. And it has also been, on the other hand, hypothesized that dysbiosis of the gut microbiome may lead to a pro-inflammatory status, resulting in higher levels of pro-inflammatory cytokines that may lead to sleep disruption. So, as mentioned, inconsistent results across studies with regard to the effects on gut microbiota composition. And this can be due to different study designs, different populations, length and extent of the sleep deprivation, but also timing of sampling, maybe during the day, sample size of the study, etc. Nevertheless, it is taking together also the human and animal studies. It's quite clear that sleep deprivation indeed has an impact on the gut microbiome, and it's called here at first, but is it really bad to have these kind of changes? would not be that sure about it, but that it has an impact, it's probably not a good thing. And also animal studies point towards a direction of a decrease in diversity. And that, I think, really could be considered as an adverse effect. So then looking into the mechanism, so how can the gut and the brain communicate with each other? So this is a concept. So first, I think, discussed in an important paper published in 2012 by Cryan and Deenan, and it emerged already before that. But now it's really, and there's credible evidence that this plays a large role. And I think it's very intriguing that our gut microbiota can influence our mood, maybe even how we behave. So many different factors play a role in the microbiota gut-brain communication. So there are many molecules, but also the so-called channels. And in this figure that we made here, we summarized a number of factors that are known or thought to play a role in sleep quality. So there's here inflammation and leaky gut, which would be unbeneficial. Microbial metabolites, secondary bile acids that are made from bile, short chain fatty acids, which I will get back to, hormones, neurotransmitters. There's the blood-brain barrier, which plays a role in the communication part. Circadian rhythm may be influenced via clock genes. The vagus nerve, which I will also get back to. Probiotics and prebiotics, of course. And as already mentioned, microbiota composition, but also function. It's important not to only analyze, if you see this, who are there. So I named a few taxa on the previous slide, but also what are they actually doing. So and on the next few slides, I will discuss the short chain fatty acids and the neurotransmitters. So as mentioned, the gut can communicate with the brain through several neurotransmitters produced in the gut. So for example, GABA, which is sleep promoting, serotonin, so I also put as sleep promoting, but it's actually also being debated, and dopamine and histamine, which are more promoting wakefulness. And also here, it's not really limited to the list here, but to name a few examples. That GABA can be produced by certain lactobacilli and bifidobacteria, and also serotonin can be produced by microbiota members, for example, by E. coli, but it's also produced by cells in our gut, so the enterochromaffin cells, under influence of the microbiome. Dopamine, for example, by bacilli, E. coli, Lactobacillus plantarum, and histamine by also on the other hand by certain lactobacilli. So it's also about the balance, probably, if a lactobacillus can produce GABA, but on the other hand also histamine. So in the end, what would be the balance in the effect on sleep? And also, I think important to mention, although cortisol, as a famous stress hormone, it cannot, as far as we know, not be produced by microbes, but we also know that probiotics can lower cortisol levels. Then the short-chain fatty acids, they are produced by the gut microbiota, mostly from undigested fiber, and they are associated with a large variety of health benefits. And the most abundant ones in the gut are acetate, butyrate, propionate. And they are important for a good gut barrier integrity and then a large variety of health benefits. And in contrast to other metabolites, the role of short-chain fatty acids in sleep is most thoroughly studied. And it has been shown that the short-chain fatty acids have sleep-promoting effects. And this is most evident, as shown by oral gavage in animal studies. But also in a human trial, the stress response was reduced after oral supplementation of short-chain fatty acids. So also a quite promising result. And so I mentioned here the short-chain fatty acids as a group, but the animal studies may also point a little bit more towards a positive effect of particular butyrate. And I think also important to mention or to realize is that short-chain fatty acids measured in the feces reflects the net of production and consumption by the microbiota and also of absorption by the intestinal epithelial cells. We really use it also as a fuel source. And this flux of the short-chain fatty acids really complicates the interpretation of fecal short-chain fatty acids levels. So, for example, if you measure the short-chain fatty acid level in the feces in a human trial. So then a bit about the proposed pathways or working mechanisms. It's quite a lot, and it's probably even more than what is mentioned here. So there is regulation of secretion of gut hormones from the enteroendocrine cells. It can activate the vagus nerve, which I will get to in a minute. Improvement of gut barrier integrity, as mentioned, but it has also been associated with indeed good sleep quality if you have a gut barrier integrity. And inhibition of the production of pro-inflammatory cytokines and even influence of the peripheral clock gene expression, which in the end may have an impact on circadian rhythm. So then I would like to move into the communication channels. So most of the neurotransmitters that are produced in the gut are actually unable to pass the blood-brain barrier. So there must be another route, and there's a large role for the vagus nerve. It is the fastest and most direct route that connects the gut and the brain, and it's also it's the largest nerve in our autonomic nervous system. And it can be activated by neurotransmitters, so for example, GABA, serotonin, but also by pro-inflammatory cytokines and microbial metabolites. And it has been shown in studies with vagotomized mice that this role is really important. So when you cut the vagus nerve, it was shown that although in normal mice, a probiotic could induce alterations of the GABA receptor expression in the brain, this did not happen if you do the same with vagotomized mice. And also after LPS injection, in normal mice, it induced depression and it exerted a pro-inflammatory response. But this was not the case in vagotomized mice. And many questions remain to be answered on the exact mechanisms here. So in this figure, it goes also a bit beyond the scope of this presentation, but to summarize a bit, so here we have, for example, the neurotransmitters, the short-chain fatty acids, the enteroendocrine cells, and there is part of the systemic circulation, but then also the role of the vagus nerve here. And what I also would like to mention, but not go into detail, is also the hypothalamic pituitary adrenal axis that also plays a role in this communication. So while we are at mechanisms already, I would like to make the bridge to the pro and prebiotics and postbiotics. So here we summarized in this figure that will probably be published soon what the mechanisms are thought to be involved in the modulation of sleep by probiotics, prebiotics, and postbiotics. So here, if we have pro, pre, and postbiotics, they contribute to a healthy gut microbiome, good short-chain fatty acids production, which will then lead to a good integrity of both the intestinal wall, but also of the blood-brain barrier. The short-chain fatty acids can modulate the immune response and this way have positive effects, but also the endocrine response, so for example, lowering cortisol, modulate also the vagus nerve stimulation here via the neural response together with the neurotransmitters that in the end is also really, the balance is important here. There are microbes that can produce neurotransmitters, but there are also pathways that degrade neurotransmitters. And then here for the circadian regulation, also short-chain fatty acids have an effect as mentioned, but also the secondary bile acids. So the contribution of each route or pathway remains to be investigated further. So what's really contributing most to the effect on sleep quality? And now the question is, knowing this, is there actually evidence for a positive effect on sleep of pro, pre, and postbiotics? We've performed a narrative review, and we focused on trials with healthy subjects. So, for example, no subjects with inflammatory bowel disease or depression whatsoever. And those studies studied the effect on sleep quality, including stress and/or anxiety. Most trials studied the effect of classic or traditional lactobacilli and bifidobacteria strains on improving sleep quality, stress, and anxiety. And specifically on sleep, there were 10 trials included in our overview on pro and postbiotics. And overall, we found that several of those have indeed a sleep improving and maybe also a stress reducing effects. So there were positive effects on sleep on, for example, the strains, formulations mentioned here. However, it was so for some other strains, it did not have an effect, but it can also have many reasons, which I will also get back to. So the results of the various trials are inconsistent, in which many factors can play a role. I would like to move to the prebiotics. So they have been studied on a much smaller scale than pro and postbiotics for sleep. So there are only 2 trials included in our overview. Galacto-oligosaccharides, but not fructo-oligosaccharides supplementation was able to lower the cortisol levels in the blood after waking up. And it also improved self-reported stress, but there was no significant effect on sleep quality, but still promising that it can reduce stress symptoms. And it is known that prebiotics, such as the galacto-oligosaccharides, can stimulate bifidobacteria and short-chain fatty acids production. So this is likely part of the mechanism behind the effect. And one recent trial that is not included in this overview, but which I would like to highlight because we were involved in the study execution, and it also had a nice result. So it was a human trial with the dairy-based prebiotic supplement performed by Friesland Campina, Philips, and also NISO. And sleep quality improved as measured by a decreased PSQI score. And this is based on a questionnaire which is called the Pittsburgh Sleep Quality Index. And in the subjects that really met the criterion of a baseline PSQI score of larger than 9, so larger indicates worse sleep quality, it was improved by this intervention. And we also found that bifidobacterium relative abundance increased in the treatment group and not in the placebo group. It's also interesting to see that in the subjects that reported an improvement in sleep, they had a relatively low bifidobacterium abundance at baseline. I think good to mention that besides GOS, there was also whey protein, vitamins and minerals and tryptophan in this supplement, so that may also have contributed to the effects. So overall, we conclude that several pre and postbiotics have sleep improving and stress reducing effects, although the results from the human trials are not consistent, which can be explained by differences in study populations, so for example, age, geographical location, and therefore also microbiota composition, gender, size of the study. Also, durations of the treatment was very different, could vary to a few weeks to maybe a few months. The type of the treatment, so probiotic, postbiotic, or prebiotic, that's of course different, but also within the probiotics, the one strain, of course, is not the other. So it can be really strain specific here. The dose of the treatments were different, and also the timing of administration during the day. So this was actually most often not controlled, but we could imagine that it could have an effect if you give it, that it needs some time to exert an effect. If you give a probiotic in the morning, maybe you can better sleep at night, or do you need to take it in the evening? And this may differ for a postbiotic or a prebiotic. So that would also be one of our recommendations to in the studies to standardize the timing of administration, to control it. But first, we need also to study whether timing of the administration really matters, if it is indeed more important to take it in the morning or the evening, for example. Although not that clear from our comparisons, it might be interesting to further explore the effects of multi-species and high CFU count probiotic treatments, and also to study more broadly the potential of post and prebiotics or a symbiotic combination, because it has been done less and might be even more stable than probiotics. And although traditional probiotics have shown to be successful, it's also very tempting to test next generation probiotics, such as Akkermansia or Clostridialis members that are typical butyrate producers. And strain selection can also be important. So screen for specific properties of strains, such as the neurotransmitter production. So we can think of why were certain strains that in the study so far, why were they chosen? Did they have already beneficial effects on other areas already just grabbed from the shelf? So yeah, we do not need to grope in the dark that much, as we nowadays have also powerful tools to do genome screening, for example. And then we can select the most promising probiotic strains based on their genetic properties. So there are generic tools for gene or pathway screening, but they rely on generic databases with limited detail. So for a study like this, looking at traits that are involved in, may be involved in sleep quality, we need a higher level of detail. And this can, for example, be done with dedicated bioinformatic modules, so-called profile hidden Markov models, abbreviated by PHMMs. By these modules, we can detect specific functions in conserved regions, so also across species. And at NISO, we could also support you in this entire process. So we could apply our custom PHMM database or publicly available modules, or build new HMMs first. Starting here in this figure also with literature research to really look for the genes or the functions that are predicted or known to be involved in this case sleep quality, but also for other health benefit areas you can do this. And in this example, we have 6 genes. So a few are associated with wakefulness and others are associated with sleep, and some are a bit in between. So based on those sequences, we can build the hidden Markov models, and in the end, these are profiles that we use to screen the genomes of, in this case, 3 candidate probiotic strains. So they have passports, but we really do not know their properties yet. So when we screen with those hidden Markov models, we know from these 3 strains what functions are present and absent. And then we need to do the interpretation. So for example, here in the blue one, we see that it is quite associated with wakefulness. So if we are looking for strains that may promote sleep, then it's maybe better to pick the yellow or the green one. And these really are predictions, so we need to further substantiate this by in vitro and in vivo studies. And for this, we could envision an in vitro screening funnel to substantiate indeed the in silico predicted health benefits, or you could just you could also start this without the in silico prediction. But at first, we could do a high throughput in vitro screening with many different candidates, and then select the most promising ones. Then look into more detail into the effects, and then really select a few that are most promising and on which you can do a clinical trial that's relatively more expensive. And also with this, at NISO, we could support you with this. We have an in vitro fermentation high throughput microcolon model in which we can study the effects on microbial composition and metabolism, and assess the production of the molecules as predicted by the in silico analysis, for example. You could also apply them directly to in vitro cell culture models with intestinal epithelial cells or immune cells or combination. And also we could combine the microcolon model with cell culture models, so then we can really look into the effect of the spent culture medium, so after the fermentation in microcolon, what it does with the cell models, in, for example, on barrier integrity or immune modulation. And then there are the clinical trials, and also might be helpful is the optimization and upscaling of probiotics including strict anaerobes. So thank you for listening, and I would like to thank my colleagues who were involved in this topic. And if you have questions, please reach out to me. And also if you think that NISO can be of assistance in your research, of course, we would be happy to discuss your project.